Assalamu alaikum everyone. My name is Mohamad Behairy. I am a third year medical student. Inshallah, today I will be going through two of your physiology lectures, endothelium and platelets and coagulation. طبعا they are very important lectures, but inshallah they are very easy. Well, I'll do my best to simplify it for you. Inshallah, this lecture will be all you need for the exam. Oh, I hope you will do great in your foundation exam. So let's get started. First of all, we're going to talk about the endothelium. But what exactly is the endothelium? Now take a look at this. This is a blood vessel. A blood vessel, what does it contain? Blood, obviously, either an artery or a vein. Now, this blood vessel is lined up by a layer of cells. These cells are called endothelial cells. They are one layer of squamous cells that line up this blood vessel. But the thing is, why would we have a whole lecture about it then? It's because this endothelium is very extremely important. It has a lot of functions, and we will talk about them in detail. For now, I want you to know that this endothelium is a layer that lines up this blood vessel, okay? Now, this is a picture in your slide, and it may seem very confusing and packed at first, but we will, inshallah, simplify it habba habba, one by one. This picture represents the functions of the endothelium. Now, the endothelium has a lot of functions, starting off with permeability. The endothelium will decide which molecules go into this blood vessel and which ones go out, because we don't want anything going inside the endothelium. And at the same time, there are some things that are necessary and crucial to go inside, such as maybe water and certain ions. So that is why one of the major functions of the endothelium is permeability, selective permeability, deciding what goes in and what goes out. Another important function is the inhibition of platelet aggregation. Now we're going to talk about coagulation in detail, but I want you guys to know that in the normal state, when a blood vessel is normal, it is not damaged, we don't want platelet aggregation. We don't want clots forming. Blacks, if the clots are going to form, blood will have a hard time passing by. So in the normal state, we don't want any coagulation because there's no need. We don't have any injury that requires coagulation. So that is the second function. The third function is helping with inflammation. Whenever the process of inflammation happens, the endothelium will secrete a lot of substances that are important to enhance this inflammation, to help the inflammation happen. Command, the endothelium itself is kind of sticky, so the inflammatory substances will be able to stick on this endothelium to do their job. So that is how the endothelium can help with inflammation. Fourth is the regulation of the vascular smooth muscle tone. We're going to summarize this sentence with two words, vasoconstriction and vasodilation. طبعا a blood vessel, if it was very small, then the blood that is passing through it, it's going to be less, which will take more time to pass through. ليش؟ لأنه it's narrow. Imagine you have a group of 20 people. You want them to walk. If we have a narrow passageway, these 20 people will struggle to walk. It will take them a long time to get from the start to the end. But if we have these 20 people in a very wide passageway, they're going to be able to pass easily. So this is a function of the endothelium, that it regulates when do we want a lot of blood to go to a certain area and when do we not want a lot of blood to go to a certain area. What do you mean by inflammation? Okay, we got this question. It's a very good question. Now, inflammation is the cell's reaction to an injury. Whenever you have an injury, tamam, whether it's a burn, whether it's a scar, whether it's a paper cut, inflammation will take place. Now, what is inflammation categorized by? By platelet aggregation, by vasoconstriction, by redness. This is why, for example, when you get a paper cut, at first you're bleeding, but if you notice after a few minutes, you're not bleeding anymore. It's like something hard has formed. This is the result of the inflammation. After the inflammation, these platelets will come together and they will prevent your blood from basically leaking out of the body. So inflammation simply refers to the process of your body reacting to a certain injury. With the endothelium of the blood cells, or any blood vessels in general, will help in that inflammation process. I hope that's clear. So last but not least, other than vasoconstriction or vasodilation, we have angiogenesis. Now, what is angiogenesis? Angio means blood vessels. Genesis means the formation of. So angiogenesis is the formation of blood vessels. Tamam, the growth of new blood vessels. So the endothelium can actually proliferate the blood vessel it's in. It can make it grow larger. It can create new ones. This is one of the functions of the endothelium. Now, to summarize, for those of you who prefer text, the endothelium is a single layer of squamous cells, which lines up the blood vessels and the lymphatics. And it has many functions, as we said, whether it's permeability. And one important thing regarding permeability that I want you guys to remember, it is selective in a way where it prevents proteins from leaving the vessel. Yani any proteins that are found in this blood vessel, let's say a red blood cell, these proteins will not be allowed to leave the cell. Well, there is one major reason. Proteins in the body are like magnets. They attract water, they attract ions. In the blood vessel, we need these ions, we need this water. That's why we want the protein to be inside the blood vessel, so that it can attract the water inside, so it can attract these ions inside. If this protein was not inside the blood vessel, if it was outside, if we let it go outside, we wouldn't have all of these important substances going inside. Because again, many molecules will follow where the protein is. So that's why it is important to prevent the movement of proteins outside to maintain the oncotic pressure. As we said, it also regulates the immune responses and inflammation, vasoconstriction, vasodilation, depending on what we need at a certain context. And it can also either start or inhibit coagulation. احنا قلنا in the normal phase, in the normal circumstances where the blood vessels are not injured, why do we want coagulation? Why do we want clotting? We won't have any thrombogenesis, any formation of clots. But when an injury happens, that's when the endothelium will help us form the blood clots. Last but not least, it forms angiotensin 2. Now, what is angiotensin 2? You're going to take it in more detail, inshallah, in renal. Just know that it increases the blood pressure. You won't really be asked about it. The only thing I want you to know about angiotensin 2 is that it is formed in the endothelium. That's all I want you guys to know. Now, we talked about a blood vessel. We talked about the layer of this blood vessel. We said that it's made up of endothelial cells. But we want to look at the relationship between each individual endothelial cell with the other. And that is perfectly described in this image. We have one endothelial cell on the right and another one on the left. These two endothelial cells actually communicate with each other. How? With junctions, tight junctions, adherent junctions. All of those junctions basically connect these two endothelial cells. Why do they have to connect? Because they will transmit substances from one endothelial cell to the other. And these junctions are what will decide, okay, this thing can pass through, but this thing can't. So the endothelium semi-permeability, as we talked about, is regulated by junctions. So for example, the intracellular tight junctions, tight junctions are tight, but they won't let big stuff go in. But because they are tight, they will only allow water and small nutrients. Like, what if the protein tried to escape from this endothelial cell, from the blood vessel? Who's going to stop the protein from escaping? It's going to be the basement membrane and the glycocalyx. So what I want you to know is that tight junctions are for the small stuff. But if we have proteins trying to escape, for example, that's when the basement membrane and the glycocalyx basically come and be like, nope, you're not leaving the cell. Now, I want you to know that the basement membrane is negatively charged. So why? It's because it contains proteoglycans. Now, proteoglycans are basically a mixture of proteins and sugars. Okay, and these proteins or sugars are negatively charged. There are a lot of them on the basement membrane. عشان كده, overall, the basement membrane will be negative. All right? Well, as we said, the glycocalyx will stop the diffusion of large negatively charged molecules. Last but not least, we have albumin, and I'm sure you heard of it. It's a protein found in the blood vessel and in the endothelial cells, and it is very important. Albumin is basically like a metrobus. It carries a few passengers, drops them off somewhere, carries more passengers, take them back to the starting point. It's literally like a metrobus. For albumin, what it does, it's inside the endothelial cells. It takes, for example, a certain hormone that needs to leave the cell. Albumin will carry it outside the endothelial cell. After it leaves, it will take another different hormone, which will bring it inside the blood vessel. This is the function of albumin. With this process is called transcytosis. So please remember this about albumin. Now, let's talk about, okay, we got a question. Can we go over the slide again, please? Yes, of course. Which slide exactly, though? Wait. Is it this one? Or the one with all of the functions of the endothelium? The one after. All right. So this one. All right, we can definitely go through it again. طيب. Between these endothelial cells that we're talking about, there are certain things that connect them. Why do we want to connect the endothelial cells? Let's say, for example, we want to take water from one endothelial cell to the other endothelial cell. There has to be like a bridge between these two cells so that the water can go from left to right. Does that make sense? But what are these bridges? They are the junctions, the basement membrane, the glycocalyx. Each one of them has a different function. For the tight junctions, لأنه they're tight and small, they will only allow the stuff that are not heavy, that are small, like water, nutrients, things like this. But what about the proteins, the big stuff? These will be mediated by the glycocalyx specifically and the basement membrane. وبعدها we were like the basement membrane is negatively charged. Why? لأنه the basement membrane has a lot of sugars and basically carbs, which are negative. That's why they make the basement membrane negative. Last but not least, we have albumin, which is a protein. Albumin, as we said, it's a metrobus. It picks up stuff, take it outside of the cell, picks up other stuff to bring them inside the cell. Well, this process is called transcytosis. Is that clear? Can you recap what you said until now and then get the first few points? The points in this slide? Well, a different slide. The blood vessels one. All right, no worries. طيب, just to recap what we said. In a blood vessel, we have something that layers it. It forms, يعني خلينا نقول a layer here. It's basically the border of the blood vessel. These are endothelial cells. And here we have an endothelial cell, here we have an endothelial cell, here we have an endothelial cells. All of them, they line up the blood vessel. Why are they important? They're important because they have many different functions, as we talked about here. They basically decide what goes in, what goes out. They either narrow the vessel or dilate it, which is called vasoconstriction and vasodilation. They create something called angiotensin 2. All right. وبعدين we talked about the relationship between the endothelial cells. يعني هم endothelial cells قاعدين جنب بعض. They are next to each other. طيب, if we have a certain substance that we want to move from endothelial cell number 1 to endothelial cell number 10, how can we do it? There has to be a bridge between them. This bridge is the tight junctions or adherent junctions. These junctions, look at them. They are literally like bridges between the endothelial cells. If we have water here, this water will cross through the tight junction to go to the other endothelial cell. This is all I want you guys to know, the relationship between the endothelial cells. Then we talked about albumin. What is albumin? Albumin, for example, is found in this endothelial cell. It will take water from inside, for example, which will go outside, which will release the water. Then it will take, for example, glucose, which will go right back in. So albumin is basically a transporter. It takes something out the cell, then goes back in with something else. It basically helps in transportation, nothing more. Is that fair? Okay, alhamdulillah. طيب, now let's move on to this. We said the endothelium also helps in inflammation. Basically when we have an injury, this endothelium will help us to react to this injury, to stop the injury, to prevent its side effects. Now, I want you guys to know, inflammation has many steps, and you don't have to go into the detail of these steps in this lecture. I just want you to know which part of the endothelium helps which cell. For example, إحنا in the inflammation, we have cells called neutrophils. These neutrophils are responsible, for example, for killing bacteria and pathogens and stuff that come from outside the body. These neutrophils go through 3 steps before being able to work. They're the rolling phase, then they are arrested, they stop, then diapedesis, which means they go inside the site of injury and they start doing their business. What I want you to know is that each one of these 3 steps has a certain molecule in the endothelium that helps it. But when this neutrophil wants to roll on the endothelium, what's going to help it roll? P-selectin and E-selectin. I just want you guys to make this link in your brain. Rolling is P-selectin and E-selectin. Arrest is ICAM and VCAM. And diapedesis is PCAM. Now, this isn't very important right now for you guys, but they really could ask a question saying like, okay, which of the following molecules in the endothelium helps in the rolling phase of inflammation? you're going to choose obviously either P-selectin or E-selection, whichever is in the choices. They could ask you which of the following molecules in the endothelium helps in diapedesis. Here you would choose PCAM. Just want you guys to know the idea that I want you to know from here is that in the endothelium there are a lot of substances that help the neutrophils or the inflammatory cells do their business basically. Integrins also help, by the way. Integrins also help, and they are found in the endothelium. But I don't want you guys to remember every single one of them. I just want you guys to know the important ones. اللي هم إيش؟ P-selectin, E-selectin rolling, ICAM, VCAM arrest, PCAM diabetes. Obviously, integrin is very important in inflammation, and it is found in the endothelium, in fact. Like, could they ask a question with both molecules? How would we differentiate? لا, they won't. If they ask you a question about rolling, you will only find either P-selectin or E-selectin in the choices. There is absolutely no way that they're going to give you both of them. Tamam? We got another question, which is, could you tell us what they stand for? If there is a mnemonic to remember the main ones. طيب, remembering the main ones. I'll try to give you a mnemonic or a way to remember. Now, when a cell wants to cause inflammation in the endothelium will not allow every single cell to roll on it. That's why we have the selectins, because they select what do we want to roll and what do we not want to roll. But when the neutrophil comes in, the endothelium will be like, okay, you can roll, you can do your business. How will we do that? With the selectins, P and E selectins. That's how you could remember rolling for P and E selectins. Now, for the rest, صراحة, I don't think there is really a mnemonic or a way to know them. But just know in the diabetes, which is the last step, will have only one thing, اللي هو PCAM. But for you guys to remember it, I made a table. Hopefully this table will help you. Oh, not a table, a simple list. Enrolling is P and E selectin. Arrest is ICAM and VCAM. Diapedesis is PCAM. Well, guys, I don't want you to really hyperfixate. You can think of rolling like a sport, so PE like in school. Absolutely, absolutely. You can think of it this way, any way for you to remember. But again, this is not the main focus of the lecture. I just want you guys to at least know them, read them. But don't waste your time knowing exactly what ICAM is and VCAM is. You will not be asked about these details. The only thing you need to know is the endothelium has substances that helps in inflammation. نقطة السطر. All right. Now, let's move on to angiogenesis. مو إحنا قلنا angiogenesis is the growth of new blood vessels. Angio is blood vessel and genesis is growth. The endothelium can help us grow new blood vessels أو enlarge existing blood vessels for different reasons, تمام? But how do they do this? For the endothelial cells, they can stimulate the growth of new blood vessels by releasing VEGF, vascular endothelial growth factor, which makes sense. Vascular in its related to blood vessel, endothelial, it comes from the endothelium, growth factor, it's for the growth. For vascular endothelium growth factor, otherwise known as VEGF, this is like a protein, a factor, it's a protein. When the endothelium releases it, we're going to have growth of blood vessels in certain contexts. For example, when we have low oxygen, our cells that are injured, these endothelial cells are going to release this VEGF, the vascular endothelial growth factor. Why? Because we want more blood vessels. Logically speaking, guys, let's say a group of cells don't have enough oxygen. وإحنا we know when the oxygen comes from the blood vessels. They contain the oxygen. But when we have a place with little oxygen, with little blood vessels, the endothelial cells are going to react. They have to save the cells before dying from a lack of oxygen. So what are they going to do? They're going to release this VEGF. Well, this VEGF will cause an increased amount of blood vessels. Now, this increased amount of blood vessels can supply this area with flow oxygen with oxygen. I hope that idea is clear. And if we have a place with not a lot of oxygen, let's say here, the endothelial cells that are here are going to release VEGF. So they can make more blood vessels to help oxygenate this area. تمام? Now, a very important concept is that in cancers, we have a big cell mass. You guys know how tumors are big. They are outgrowths. تمام? The problem is when a tumor starts getting bigger and bigger and bigger, this thing that is getting bigger is something that is pretty much, they are mutated cells. تمام? For this region becomes hypoxic. لأنه this tumor growth is causing a decrease of oxygen. إنه ترى by the way, tumors are living cells. The more they grow, the more oxygen they need. But when they grow so much, there will be a lack of oxygen. So what happens? The endothelial cells of this tumor will release VEGF to get more and more blood vessels to supply the tumor. That is why when a cancer spreads or starts, it gets bigger and bigger and bigger. Because as the cancer gets bigger, it demands more oxygen. ولأنه it demands more oxygen, we will have more VEGF and more blood vessels to grow. This will cause the tumor to get bigger and bigger and bigger. So we need a VEGF inhibitor in some cancer treatment? Exactly. This is the exact link I want you guys to make from this slide. And sometimes we want to inhibit VEGF. When we have a tumor. Perfect, ya Ahmed. This is the perfect خلينا نقول conclusion you can get from this slide. I hope this concept is now clear. Now, regarding vascular tone, okay, wait, we have a question. Is it the Avastin? Yes, it is this one, Avastin. Now, in your exams, guys, they will never ask you about drugs, pharmacological drugs. You will deal with this, inshallah, in year 2, year 3. I just want you guys to know the concept. And when we have a cancer, it's getting bigger, it needs more oxygen, we will have more VEGF, which means more blood vessels, which will get even bigger. That's why sometimes we would want to inhibit it. That's the only thing I want you guys to know. I just want you to know what VEGF is. Vascular tone, it refers to vasodilation or vasoconstriction. The endothelium, if it wants to dilate, it will release certain substances that will cause this vasodilation. If it wants to constrict, then it will also release certain substances that will constrict the blood vessel. Tamam, depending on when we need to dilate and when we need to constrict. Now, I want you guys to memorize these. Nitric oxide or endothelium-derived hyperpolarizing factor, they cause vasodilation. Or a way to remember it is hyperpolarizing, hyper, a lot, big. We want to do vasodilation. Endothelin, on the other hand, will cause vasoconstriction. All of these three substances موجودين في الendothelium. When the endothelium decides it wants to dilate, it's going to release nitric oxide or EDHF, which is endothelium-derived hyperpolarizing factor. If it wants to vasoconstrict, then it will release endothelin. These molecules are what will regulate the vascular tone. We will come back to this later, but nitric oxide not only does vasodilation, but it also inhibits coagulation with prostacyclin. We will go back to these later. Oh, this is prostacyclin. Prostacyclin, it inhibits coagulation. It's like nitric oxide. Tamam? We will talk about them later on. Now I just want you to know in nitric oxide or EDHF, vasodilate, endothelin, vasoconstrict. This is more than enough for now. We got to a checkpoint. Does anyone have any questions so far? By the way, everything we just did is everything you need to know about the endothelium. You don't need to know anything else, inshallah. This is everything about the endothelium. Does anyone have any questions? Okay, seems in no, we have a question. Can you go back to the previous slide? Okay, of course. One moment. Guys, a question. Now we'll start with the coagulation. Now we're going to talk about platelets, the second part of the first lecture. Guys, don't worry, we will be done in no time, inshallah. These two lectures, they might seem difficult to complicated. This PAL session is everything you need for these two lectures. I made sure to cover everything important. Even the practice questions in this PAL session, والله العظيم يعني أنا حلفت, they will be the same questions you're going to get in the exam, inshallah. I made sure to cover all of the major ideas. This slide, guys, I just want you to know one thing. There are certain things in the endothelium that cause vasodilation or certain things that cause vasoconstriction. I gave you some examples. I don't necessarily want you to memorize every single example, but what I want you guys to know is إنه في substances that do this or others that do that. The ones that do vasodilation هم nitric oxide or endothelium-derived hyperpolarizing factor. These are just examples. You don't need to memorize every single thing, every single example. Endothelin, we will come back to it later in the coagulation, but I want you to know it causes vasoconstriction. So, ma'am, that's everything you need to know here. I just want you to know the endothelium can vasodilate or vasoconstrict. Now, going back to the inflammation, all I want you to know is the endothelium helps in the process of inflammation. How? Through these molecules. P-selectin, E-selectin, I-CAM, V-CAM, P-CAM. Again, I don't want you to know what exactly they do or memorize them perfectly. I just want you to know, for your own knowledge, for rolling, we have P or E-selectins. وفور arrest ICAM وVCAM وdiapedesis PCAM. You don't need to go into the details of each one. If you want to know, just for the sake of knowing, when an inflammatory cell comes, it starts by rolling, بعدين it's arrested through the integrins, بعدين diapedesis, which is basically this inflammatory cell going into the site of injury. So it can attack whatever is causing the injury. You don't need to know all of this. I just wanted to give you a clear full picture. What I want you to know is إنه the endothelium helps with inflammation. It's good to know this. إنه والله rolling is done by POE selectins, arrest by ICAM, BCAM, diapedesis by PCAM. I don't want you to go into any more details. Wallahi, yani, you won't need it right now. We got a question. Of course, you're welcome. Right. Now I have a question for you guys. I want someone to open their mic, read the question, give us an answer. Well, come on, everyone else that is watching the past session, feel free to throw in what you think the answer is. I just want a volunteer to read the question for us. Please, open your mic, maybe. Feel free.
Okay, so a 50-year-old woman undergoes surgery to remove a large abdominal tumor. Histologic findings show that the mass contains a large number of blood vessels. An increase in which of the following is most likely responsible for the hypervascularity of the tumor?
What do you think the answer is?
It's gonna be VEGF, like as you said before. The tumor needs oxygen, so that's going to lead it making the vessel larger
Exactly.
to get nutrients from the vessel, and it does that by releasing VEGF. So C should
Exactly.
be the correct.
Yep. Thank you so much, Munib. That is the correct answer. Wa mashallah alaykum. Everyone in the chat got it correct. It is VEGF. By the way, guys, I wanted to tell you this after we're done with the question. Inshallah, this could motivate you. But this is a question that we got in our foundation final. For the fact that you got it correct means that there is absolutely no reason why you can't get the other questions correct. Tamam? For this was a question from a real exam. Wa mashallah alaykum. You guys got it correct. Now let's move on. I'm sorry, could you repeat what causes hypervascularity? Okay, لا. Nitric oxide was for vasodilation, to make the vessel wider. Hypervascularity, what does it mean? Hyper means a lot, vascularity means blood. The hypervascularity means a lot of blood vessels. وإحنا قلنا إنه VEGF creates blood vessels, which means we will have more blood vessels, obviously. When cancers, the more big they get, the more oxygen they'll need. Tamam? But they will have a lot of VEGF, a lot of new blood vessels, hypervascularity. That's why cancers get bigger and bigger and bigger. Okay, what was EDHF again? Okay, we can go back to the slides. It's endothelial-derived hyperpolarizing factor, just so you guys can see it. This endothelium-derived hyperpolarizing factor. I don't want you guys to know any details about it. Just know it's a vasodilator. Bas. I just want you guys to know the main concept. No need to memorize and nitpick the details, يعني. Inshallah. Right, now we finished the first part. Now let's move on to platelets. Platelets are much easier. I think you guys took this in your hematopoiesis lecture. The one thing I want you to absolutely memorize and remember from this is how do platelets come from megakaryocytes? Megakaryocytes are going to shed. They're going to break down into small pieces. These small pieces are going to end up being our platelets. This is all I want you to know from this. Megakaryocytes will shed, and this shedding will be the platelets. Tamam? Now, what are platelets? They are cells with no nucleus. They are produced by the budding, as we said, and shedding of the megakaryocytes. طبعا, what is their growth controlled by? A hormone called thrombopoietin. Well, I'm sure you guys took this in the hematopoiesis lecture. You know when it regulates how much platelets we want to grow. Yani if we have مثلا, a lot of thrombopoietin, a lot of megakaryocytes are going to shed and bud into platelets. If we don't have a lot, we won't have a lot of platelets. طبعا, when would thrombopoietin be a lot? We need a lot of platelets when we're bleeding, for example. That's when we need a lot of platelets. The small segments of the fat cell megakaryocytes and regulates via thrombopoietin. Absolutely, exactly. This is the perfect way to represent it. Megakaryocytes will shed into platelets. This is regulated by thrombopoietin. تمام؟ طبعا This is not very important. I just put it for completion's sake. And with the cytoplasm of the platelet, again, like any cytoplasm of any cell, it will contain granules, serotonin. In this case, an important thing, von Willebrand factor. We will talk about it in the coagulation segment. But what I want you to know from this is those two things in red. Glycoprotein 1b, glycoprotein 2b, 3a. Tamam? Now, when we have platelets, what do we want platelets to do? May we have a question, it seems. Can you please send the PPT here so we can revise it before the TPL? طيب. Yahya, I think you're here in the session. If you could please send it to them, wallah, I would appreciate it. If we found a not normal platelet, we blame thrombopoietin. لا شوف, this isn't how it works. If we have an abnormal platelet, it would be related to something else. In thrombopoietin, all it does is it gives the megakaryocyte the green light to become a platelet. It gives the approval for the megakaryocyte. Yalla, become a platelet. Abnormal platelets are not really a result of that. GND pathway. Okay, whoever asked for the slides, do you know how to access the link for the PAL slides? Because everything is found there. ويحيى بالله, if they don't know how to, if you can send the link here, I would appreciate it. طيب, but what are glycoprotein 1b or 2b3a? Let's explain it in this very simple way. تمام? I injured my hand. Hello, I started bleeding. What will platelets try to do? They will try to stop the bleeding. How? Two steps. Number one, the platelets have to stick to the place where I'm bleeding. صح? Well, that's called adhesion. For the platelets will stick to wherever I'm bleeding. Second thing is aggregation. We don't only want one platelet to stick, we want a lot of platelets to stick. The process of how platelets work is divided into two. We have the platelets sticking to the area of bleeding. We have the platelets aggregating on each other. So adhesion and aggregation. Can someone send it here? I'm not in the 18th batch group. But guys, please, if anyone can volunteer to send the link of the GND drive so they can access the slides, wallahi, I would appreciate it. Okay, perfect. خلاص, someone sent it. Thank you guys so much. Thank you. طيب, why was I talking about adhesion or aggregation? لأنه every process of these has a certain glycoprotein that makes it happen. Glycoprotein 1b, من اسمها, it is 1b, it is number one, which means it is involved in the first step. What is the first step? Adhesion. But glycoprotein 1b is for adhesion. Glycoprotein 1b helps the platelet stick to where I'm bleeding. Glycoprotein 2b3a, خلاص, it's 2 and 3, the step after step 1, it's going to be aggregation. First we stick with glycoprotein 1b, then we form together, we aggregate, نصير أكثر وأكثر. That's why glycoprotein 2b3a. You guys are going to be tested on this a lot. One of them will be in the practice question you're going to see soon. طبعا there are certain clotting factors. Come on, they are found in the platelet. I don't want you to remember in the 1 or 5 or 8 are on the surface of the platelet. Just know in general, the platelet has clotting factors. طيب, adhesion or aggregation, we talked about them. Let's talk about them again in a way you can visualize it. We have an endothelium that is damaged. The platelets will bind to the von Willebrand factor. We will talk about the VWF in detail back way. Just for now, know and whenever there is an injury, whenever the endothelium is damaged, whenever the first layer of endothelium is damaged, it will expose something called VWF, von Willebrand factor. With this, على طول will bind to the platelets. تمام؟ بعدها, we will have synthesis of thrombin. What is thrombin? It's a hormone that activates platelets. For the first few platelets, will make thrombin. With this thrombin, will make even more platelets, so even more platelets. Well, this is what we call aggregation. Hello? So we have a damaged endothelium. The platelets على طول will bind to the place of injury, على الفون Willebrand factor. Well, this platelet will realize that on its own, it can't stop the bleeding, but it's going to have to call its friends. How? By secreting thrombin. This thrombin will get more and more platelets to be activated. Well, then they will aggregate. This is adhesion or aggregation. But don't forget, adhesion is done, it's the first step, so it's going to be done by glycoprotein 1b. Aggregation is the second step, that's going to be glycoprotein 2b and 3a. Tamam? Now, we have a question. Does it secrete thrombin or لا, it secretes thrombin. Okay, we'll go back to the slides. ولا يهمكم. The thrombin will release once a platelet binds to VEGF. Not necessarily, but usually we only want to secrete thrombin when we have an injury, damage to the endothelium. So that's why in most cases we're going to have this thrombin released after the injury. Collagen is very important, obviously. Under any endothelium, under any cell, you're going to find collagen. But collagen is not what's important here. What's important is the von Willebrand factor. You will not be asked about collagen in the exam abadan. As collagen is simply connective tissue. What we care about is the von Willebrand factor, the VWF. ليش؟ لأنه this is what's going to hold the platelets, help it for adhesion. After that is when we have thrombin, or all of these aggregations. 1B, 1B. We have 1B or 2B, 3A. 1B is for adhesion, first step. Second step, aggregation, 2B, 3A. Exactly, Ahmed just said them perfectly. No, wait. Yeah, okay, exactly. Glycoprotein, 1B, collagen, 1B. 1B is also for adhesion. Let's go back to it عشان ما تلخبطوا. 1B, adhesion. 2B, 3A, aggregation. This is all I want you guys to know. You're going to see it in a question, right? طيب, platelet storage granules. طبعا I just want you to know in the platelets we have a lot of things that can be found in the granules of the platelets. مثلا ADP, ATP. أكيد the platelet needs energy. There are also lysosomes. What are lysosomes? They basically break down waste or unneeded substances. We also have clotting factors. قبل شوية we said إنه in the cytoplasm of the platelets we have granules or serotonin or factors. This is just like a table summarizing them. You don't need to memorize every single thing. Just know that there are granules or clotting factors or stuff like that. تمام? Now, what are platelets activated by? As we said, von Willebrand factor, the most important thing. It will immediately bind to the platelets or begin the adhesion process. Number two, AGP. Why AGP? إحنا, when we have an injury, we start bleeding. What does this mean? How does this blood come out? It comes out from a blood vessel that died. What are blood vessels? Right, red blood cells. Red blood cells are blood vessels, where they contain blood. But when the red blood cell basically explodes or dies, the blood will come out, but كمان AGP will come out. With this AGP will tell the platelets إنه we need you. Platelets will be deactivating, sitting, having a cup of coffee, everything is normal, no one is affected. Then it's going to see random AGP molecules flowing through. The platelet will be like, okay, there are AGP molecules. This means that there are red blood cells that are damaged. Well, that's when platelets get activated. As we said, كمان we have thrombin. With thromboxane, we will talk about it in detail with coagulation. But the one we talked about were thrombin and von Willebrand factor. طيب, let's go to the next checkpoint. Do you guys have any questions? كده احنا officially we finished the first lecture. Okay. طيب, I have a question for you guys then. Oh wait, شكله في question. What's the difference between thrombin and thromboxane? Proof, not very big differences, but thromboxane, the main thing thromboxane does is for the coagulation that is after platelets. Vasoconstriction or clotting factors. We will go into it, intrinsic, extrinsic pathway. Thrombin's main task is to get more platelets. Thrombin's main task is to get more platelets. Thromboxane's main task is to mediate the whole process of coagulation, intrinsic, extrinsic factors, ما أعرف إيش. Summarizing the steps of adhesion and aggregation, of course. طيب. First of all, from this slide, I just want you to know that there are things that activate platelets, most importantly, von Willebrand factors, AGP from the damaged RBCs, thrombin with thromboxane. This is what I want you guys to know from this slide. تمام? What do the hydrolytic enzymes do? طيب, let me tell you. Let's say مثلا we have a cell, or somehow a bacteria was able to go inside this cell. But this bacteria is not strong. It's just one bacteria. It doesn't need a whole inflammation. What's going to happen? And then these lysosomes, these hydrolytic enzymes, will chop them. As simple as that. For hydrolytic enzymes are like the washroom. Wash off things we don't need. حرفيا. It's found in practically every cell. تمام? Now, going back to adhesion or aggregation, I'll explain them like this. I'll explain them in a very simple way, without needing the slides. I got injured here, تمام, I started bleeding. The first step will be that a platelet will come and bind to the place of this bleeding. Why? Because it wants to stop the bleeding. But this platelet will realize, and on its own, it can't do much. That's why it's going to call even more and more platelets to help it stop the bleeding. And that's aggregation. The adhesion is when a few platelets come and stick to the area of bleeding. Aggregation is when multiple platelets come and aggregate together to stop the bleeding at once. That's everything I want you to know with regards to adhesion or aggregation. Well, I also want you to know in glycoprotein 1b, adhesion, glycoprotein 2b, 3a are responsible for aggregation. This is very important one. How do we get AGP? Obviously, turn AGP into ATP. Exactly. Ahmed just summarized it perfectly. بس إنه exactly. Whatever Ahmed said, it is absolutely true. إنه when the red blood cell will die, it's going to release AGP. طبعا AGP is in a constant cycle of becoming ATP and back to AGP. But when the RBC dies, it will release the AGP it had. Without the signal the platelet needs to know when there is damage. تمام? طيب, I want someone to volunteer to read this question and answer it. Everyone else, feel free to type in the answer in the chat. Okay, mashallah, everyone's already answering. خلاص, okay, everyone is correct, mashallah. I'll read the question. In patients with autoimmune thrombocytopenic purpura, purpura are basically red spots of bleeding. You don't need to know them. It's just for you to understand. Antibodies are made against glycoprotein 2b3a. Which choice below is the mechanism for the bleeding in these patients? MashaAllah, all of you got it correctly. It is the inhibition of platelet aggregation. Once again, guys, this is a question that was found in one of the past finals. You answering this question correctly, mashallah, means there is no reason that the other questions will be hard for you. This is the kind of question you will get in your final. Well, because you guys, mashallah, were able to answer this very easily, it means that you shouldn't worry, inshallah. And yes, adhesion is 1B. I will explain. I will explain in all simplicity. Tamam? The question is asking, antibodies are made against glycoprotein 2b3a. For which of the following is the mechanism of bleeding? Yeah, we have a problem in 2b, or this problem is causing excessive bleeding. Tamam? Here it's asking which of the following mechanisms is what's responsible for this extra bleeding. احنا قلنا قبل شوية in the slides. Let's go here. In 1b, glycoprotein 1b is responsible for adhesion. Glycoprotein 2b3a is responsible for aggregation. Here in the question, they're asking about glycoprotein 2b3a. Plus, the moment you guys see 2b3a, the answer is going to be platelet aggregation. Is that clear? This is basically not really a critical thinking question. It's a recall question. The question is trying to test if you know the function of glycoprotein 2b3a ولا لا. واحنا قلنا قبل شوية 1b adhesion, 2b3a aggregation. So when you see 2b3a in a question, you're going to choose inhibition of
Like
platelet aggregation.
they think.
What was that?
My bad. I was on mute.
Okay. No worries. Feel free. Right, I hope this was clear. Are most MCQs clinical or recall? To be honest, a lot of them are recall. A lot of them require you to know the links between two things. This is what I'm trying to do in this past session. I want you guys to learn how to make links. For example, I was telling you guys, I want you to make a link between 1B and adhesion, or 2B, 3A and aggregation. But understanding is very important. If you don't understand, you won't be able to answer. But at the end of the day, what's the point of understanding if you don't remember which one is 1B or 2B? Memorization is always important in medicine, especially during your first year. There will be a lot of questions like this. And they're trying to test whether or not you memorized which factor, which glycoprotein, like in this question. But yeah, but don't worry, it's nothing too complicated, nothing too hard. The fact that you guys answered the first two questions I had for you in all simplicity means that the FND final will be very easy for you, inshallah. I don't think you're gonna get questions harder than this, Salah. طيب, first lecture is over. Now we're going to go to the second lecture, which is coagulation. Inshallah, we won't take long. Might seem complicated. Yes, it is a very heavy topic, but it's very fun. Well, I'll make it as simple as I can for you. Again, with all the questions you need to ace the fine. But first of all, let's revisit a concept we talked about. In the endothelium, normally it doesn't want clotting. It doesn't want thrombogenesis. Why do we need clotting and coagulation when the endothelium is normal, when there is no damage, when everything is normal, we're not bleeding? ما نحتاج. طيب, but now let's talk about how this happens. How is the endothelium maintaining this anti-thrombogenic state? تمام? It maintains it with multiple substances. With these substances, now I want you to memorize them. Yani these are important. First, we have things that inhibit the activation of platelets. Yani things that stop platelets from even forming or activating. What are they? PGI2, which is prostacyclin, nitric oxide, and ADPase. Guys, مو احنا قبل شوية we said إنه when there is ADP, the platelet will get activated. طيب, it makes sense in when ADPase, something that converts the ADP, that breaks it down, will not make the platelet activate. صح? And if ADP activates platelets, then ADPase will inactivate the platelets. I won't allow them to activate in the first place. Nitric oxide من البداية we said إنه it's a vasodilator. Any vasodilator will inhibit the coagulation cascade من الأخير, including prostacyclin. Prostacyclin also inhibits the activation of platelets. But these three are for platelets. Then we have one thing, more than one thing, but for now I want you to know one thing that inhibits coagulation. Not the platelets, that inhibits the second part of coagulation, which is the fibrin of عارف. We're going to talk about it right now. That's going to be antithrombin. قبل شوية we talked about thrombin. Let's go all the way back. I just want you guys to make the link. We said the platelets are activated by thrombin. For antithrombin, obviously, is going to inhibit thrombogenesis. It's going to stop us from having coagulation. صح? Last but not least, is something that can't inhibit the formation of platelets or coagulation, but it's going to break down the clot. يعني احنا we can either stop at the step of the platelets or at the step of the full coagulation with the fibrin, or if we are too late, just break the clot itself. But the endothelium stops coagulation at all of these steps. What inhibits platelets? These three: ADPase, nitric oxide, and prostacyclin. These are what inhibit platelets. حتى antithrombic, by the way. طيب, what is the thing that is going to break down the fibrin in a process called fibrinolysis? Into guys, have you ever gotten like injured, for example, in your knee or your hand, which starts bleeding? Or after 4 days, you find this crusty red thing in your hand? This is fibrin. This is fibrin, tamam? This thing that is hard after a few days of any bleeding you have, this is fibrin. But it doesn't stay forever, right, guys? حتى some of us just scratch it ourselves. But if we don't scratch it, كذا ولا كذا, it will fall off on its own. That is fibrinolysis. The fibrin, خلاص, it will break down. Well, this is done by tissue plasminogen activator, which will activate plasmin, or plasmin will do it. Yes, of course, the thing that kids like to remove. Absolutely. The kids always like to scratch it off themselves. Like they do their own manual fibrinolysis. The scab, exactly. Yes, it's a scab. Absolutely. That's what fibrinolysis is about. This scab is made up of, شوف, platelets are only during the first 30 minutes. While in platelets, we will explain it now, they are temporary. You can't really depend on them. You know, when you have a leak, for example, in the sink, in the bathroom sink, you can use tape in the beginning to stop the leaking of water. But you only use the tape until the plumber comes who fixes it with something completely different. For the platelet is like the tape, it's temporary until the actual real solution comes. لأنه fibrin takes time to form. وإحنا we need to stop bleeding as soon as possible. That is why we start with the platelets. تمام؟ طيب. How does an endothelium suddenly become thrombogenic? وفجأة يصير عندنا coagulation or clotting or whatever. It's when we have damage to this endothelium. When the endothelium is damaged, when this outside layer of the endothelial cells is scraped off, the von Willebrand factor will finally show. What did we say about the von Willebrand factor? It's an activator of platelets. But the first step that makes an endothelium want to cause clots is activation of platelets. So as we said, adhesion or aggregation. So that platelets would come and bind or form this clot. صح? But as we said, platelets are temporary. They can't stop the bleeding forever. We need something stronger than platelets, and that's fibrin. How do we get and make fibrin? It's like the coagulation cascade. And we're going to talk about it. Intrinsic pathway, extrinsic pathway, common pathway. This is what's going to make the fibrin, the strong layer that's going to stop the bleeding. Tamam? The last way in which the endothelium will stop or will promote clotting when it's injured is by inhibiting fibrinolysis. Inhibiting it. يعني احنا مو we said in the normal state the endothelium doesn't want clotting, it doesn't want coagulation. When it's injured, لا, it wants coagulation. It wants to stop the blood from being lost. So I hope this concept is clear. I just want you to know, and we have two states of the endothelium. When it's normal, unaffected, it will not want to cause clots. إيش الفائدة? Why do we want coagulation? When it's injured, it will be thrombogenic. It will want to form clots. Do I need to know the factors by name? We're gonna go through every single factor in a bit, or I'll tell you exactly what I want you to know, what I don't want you to know. For now, I just want you to know in prostacyclin, nitric oxide, ADPase, antithrombin, all of these things, tissue plasminogen activator, they are antithrombogenic. They stop clotting. تمام? Or I want you to know when the endothelium is damaged or injured, the opposite will happen. As we said, the von Willebrand factor is exposed. We will have the platelets, adhesion, aggregation, all of this. تمام? As a summary, this is a table for you guys. Antithrombogenic endothelium. How is the endothelium going to stop itself from having clotting and coagulation? With prostacyclin, nitric oxide, ADPase, plasmin, all of the things we talked about. Is it enough to memorize all the TPA? TPA is enough. حتى UPA you don't really need TPA. Well, I want you to know in مو أنا قلت لكم إن there is a scab, or basically in after a few days of any bleeding, it's going to fall off on its own. How is this fibrin broken down? By plasmin. What activates the plasmin? Tissue plasminogen activator. For all of these things are trying or working their best to remove the clotting, the coagulation, for they are antithrombogenic. The thrombogenic factors are again von Willebrand factor, tissue factor. You don't need to know it now. We're going to talk about it. Or plasminogen activator, inhibitor. مو احنا قلنا plasmin breaks down the fibrin. طيب, when the endothelium wants this fibrin, it's going to inhibit the plasminogen activator. Hence why plasminogen activator inhibitor. تمام? So in normal state, platelets are just inhibited at all times by NOPG2 and ADPase, 100%. I thought fibrin is broken down by TPA. We're going to come to this, but I'll answer your question right now just to feed your curiosity. Look, tissue plasminogen activator, it's an activator. من اسمه, it's an activator. It turns plasminogen into plasmin. There are some substances in our body that are found in an inactivated form. Well, they always end with the letters OGEN, ogen. So plasminogen is plasmin that is inactivated, plasminogen. TPA, which is the activator, will bring the plasminogen and it will turn it into plasmin, which is the active form. Or plasmin, it's what's going to basically break down the fibrin. Is that clear? We will talk about this, guys, in detail. Now I'm just trying to give you a good foundation, a good base of knowledge, so we can streamroll through the concepts. But now we're going to talk about the full journey that happens when you guys bleed anywhere in your body. The first thing, you're going to have a vessel injury. As we said, then we're going to have a platelet plug. The platelets will come and they will form this plug. But as we said, platelet plugs are temporary. They can't stop the bleeding forever. They're not strong enough. They are just temporary until the fibrin is finally formed. But you might say, why do we not use fibrin in the first place? Why do we need platelets? It's because fibrin takes time to form. It's like when you have a leak in the washroom sink and you're waiting for the plumber to come. So you just block the leakage by using tape until the plumber comes and they'll fix it for you. That's exactly the concept here. What makes fibrin? We will talk about it in detail in the common pathway, don't worry. It's fibrinogen. It's the inactive form of fibrin. We will talk about it in a bit. تمام? After the fibrin finally forms, خلاص, we don't need the platelet plug anymore. Now we have the strong fibrin. With the fibrin, we'll make the final clot that will prevent and stop the bleeding. And this whole process can be divided into two parts. The part until we get the platelet plug, and the part where we don't need the platelets anymore and are now using fibrin. تمام? The first part, until we have a platelet plug, is called primary hemostasis. It's primary. It's the first reaction to bleeding. After we start having the fibrin, it's going to be secondary hemostasis. It's the second step of stopping bleeding, where we don't need platelets and only need fibrin. Primary action in a leak is a tape, exactly, until the plumber comes and uses the fibrin. Does the fibrin mix with the platelets? Yeah, absolutely. And it's not like when the fibrin comes, all of the platelets fall down together. لا خلاص, they can stay عادي. يعني they help each other. But the platelets just hold and stop the bleeding temporarily until the fibrin comes or the fibrin finishes the job. That's pretty much it. We divide it into primary hemostasis, secondary hemostasis. As we said, hemostasis is basically the mechanism of trying to lose blood. Vascular spasm is the same as vascular constriction, right? 100%, مية. Because when a blood vessel ruptures, when we have bleeding, we want to constrict the vessel to prevent blood from being lost, you know? What about the collagen fibers? Look, the only thing I want you to know about collagen fibers is that they are found under the endothelial cells. But when we have an injury, the von Willebrand factor and the collagen will be exposed to the outside. Well, this will make the platelets go crazy. And then we have to plug. We have to plug. We have to protect the endothelium. It doesn't have a layer anymore that protects it. That's all I need you to know. Of course. But it starts, as we said, by forming a temporary platelet plug, which will later be replaced by a fibrin plug. وخلاص, when the bleeding finally stops or the injury is over, we're going to lyse the fibrin and break it down. But as we said, primary hemostasis is about forming the platelet plug. Secondary hemostasis refers to the clotting cascade, which will lead to fibrin. تمام? Let's go to this checkpoint. Does anyone have any questions? Well, I hope that concept is clear. We can move on to a question. Oh, okay, Shakir, we have questions. Can you repeat the thing about collagen fibers? Okay. Can you just recap the last slides? خلاص من عيوني, I'll do that. طيب. This is the last slide. I'll explain it by using the diagram. Into guys, when you get injured, when you start bleeding, the first thing that's going to stop the bleeding are platelets. Platelets are very fast acting. على طول, they come and they form a plug and they stop bleeding. This is called primary hemostasis. تمام? But these platelets that stopped your bleeding, they can't stay forever. They're not very strong. Eventually, the bleeding will overpower the platelets. You're going to bleed again. That's why we need something stronger, and that is the fibrin. Fibrin takes time to be made. That's why we have the platelets stopping the bleeding at first. And then eventually, the fibrin is going to come, وخلاص, stop the bleeding for good. The process of making fibrin is called secondary hemostasis. The process of making the platelet plug is called primary hemostasis. Now, the person that asked about collagen fibers, look, in any cell, you have the epithelium or endothelium layer of cells. Below it, you have collagen. But obviously, when we have an injury or the endothelium is stripped apart, the collagen حيبان. It's going to be exposed to the outside. Well, this is not good. The platelets will want to cover it and protect it. This is basically it. How hard is a fibrin clot? I can tell you that it's hard enough to stop the bleeding for good. Or if you really want to know how hard it is, يعني لا سمح الله, if you ever gotten an injury and started bleeding, wait a few days, maybe a day or two كده, حتى even less, you can feel how hard it is. You know, it's a scab basically. As you said, the one that children like to basically scrape off their whatever. We need von Willebrand factor because platelets can directly bind to the exposed collagen, right? 100%. And عشان كده, guys, I told you focus on the von Willebrand factor because the von Willebrand factor is what's going to trap the platelets, allow them to bind. The same concept happens inside the body rather than the skin? Yes, but we're going to talk about what happens inside the body. But yes, the same concept. Hemostasis is in all of the body. But let's move on to a question I have for you guys. Please feel free to answer in the chat, and then we will go over it. لا, we're not finished. لسه باقي basically one more part. Let's see the answers in the chat. B. Okay. Which process below is responsible for keeping the endothelium in a non-thrombogenic state? Yani we don't want coagulation, we don't want clotting. Everyone, mashallah, got the correct answer. Synthesis of prostacyclin. This is how they're going to ask you about it in the exam. They're not going to tell you to list them or something. لا كده. Multiple choices that are wrong. One of them that's correct. Oh, mashallah alaykum, guys. You got it on spot. The one that inhibits platelets activating 100%. Okay, someone said they don't get it. طيب, I'll help you get it, inshallah. Here they're telling you which of the following is it the same as PGI2? Yes. Prostacyclin is abbreviated as PGI2. But the question is saying which one of these options is responsible for not allowing the endothelium to cause coagulation, platelets forming or clotting. تمام? فإحنا, if we go back, we said normally when the endothelium is not injured, it is antithrombogenic. And if the endothelium is not injured, why would we want clotting and coagulation? صح? We said the way the endothelium is antithrombogenic is through certain substances that are secreted by the endothelium. What are these substances? We named them in this table. Antithrombogenic, we have PGI2, which is prostacyclin, nitric oxide, ADPase, antithrombin, plasmin. Well, there are a lot of stuff that are thrombogenic. But here the question is basically saying which of the following is antithrombogenic. But until you look through the options, which option contains one of these? And that's the correct answer. That's basically it. Is the question clear? خلاص, ممتاز, of course. Right, now let's move on to primary hemostasis. We already talked about it. But now let's take it with baby steps. I got injured here, تمام? Now I'm bleeding. What is the first response going to be? Vasoconstriction. ليش? لأنه I don't want to lose blood. If I'm bleeding, I can't stop the bleeding, might as well try to minimize it. Well, that's through vasoconstriction. And if the vessels are dilated and are expanded, a lot of blood will be going through them. But if the vessels are constricted, they are narrow, not a lot of blood will be able to leave. So this is the first response to any injury or cut you get. تمام? What is vasoconstriction stimulated by? We already talked about many things, but thromboxane is one of them. We already said endothelin. Do you guys remember it? We're mentioning it again. We have a question. Would vasoconstriction not increase blood pressure so more blood is lost? Sure. Vasoconstriction doesn't necessarily increase blood pressure. Could sometimes, but not in this case. When we have an injury, يعني blood pressure, أصلا it's good to increase it. You want to know why? When someone loses a lot of blood, they become hypotensive. What does hypotensive mean? It means that their blood pressure is low. Then they're losing all of the blood in their body. But when we vasoconstrict, when we narrow the vessels, we increase the blood pressure. For the patient will stay stable. فلا, increased blood pressure does not mean losing more blood. If anything, it means losing less blood. A vasoconstriction will narrow the vessels, increasing the pressure, or not as much blood will be leaving the cell. Is that clear? As we said, vasoconstriction minimizes blood flow, and hence less blood will be lost. Okay, ممتاز. طيب, platelet plaque formation, we already talked about it. An injured vessel will release ADP, which attracts platelets. مو احنا قلنا قبل شوي انه when a red blood cell dies, it will release ADP. when this endothelial cell is exposed, we have the von Willebrand factor and collagen fibers. All of these things will attract platelets. The platelets are basically attracted by anything in this. So, then these platelets will attach to the von Willebrand factor. This is adhesion. Do you guys remember adhesion? They will attach to the von Willebrand factor and they will release thromboxane and thrombin. All of these things will cause more vasoconstriction and more platelets to come. As we said, more vasoconstriction and more platelet activation. بعدين, as we said previously, the glycoprotein 2B O3A will cause aggregation and cross-linking. Cross-linking basically means the platelets are connected to each other so they can form one plug. Is this slide clear? We already talked about everything here. But if there's anything that's unclear, tell me, I'll repeat it. And I'm trying to repeat every concept multiple times just so it's 100% engraved within you. As here we're just saying when the vessel is injured, it will release certain stuff that will attract the platelets. Platelets will release thromboxane or thrombin or these stuff, will cause vasoconstriction and even more platelets. وبعدين we will have the aggregation. GP stands for glycoprotein. It is a protein that contains glycogen or sugar. Thromboxane is released by the attachments of platelets to von Willebrand factor. Yes, that's one way you can release thromboxane. Where does glycoprotein 2B come from? It doesn't necessarily come from anywhere. It's found in the endothelium. How does ADP attract platelets? It's not necessarily the platelet will see the ADP, as I said, yalla. This means we have to form a plug. But every cell has certain antigens, certain things. It's like, you guys know how an antibody recognizes مثلا pollen grains, well, that's why an allergy happens. It's kind of the same things here. The platelet will recognize certain antigens on the ADP, well, that's why it will activate. Is that clear? I hope I answered all the questions. Okay, alhamdulillah. Guys, once again, wallah, this past session, inshallah, it will cover both Abdul Jabbar lectures, so you really don't have to go back to them. Glycoprotein 2B is similar to, but the other one is in Roman numerals. That's the only difference, Wallah, they're the same thing. Okay. Now, I want you guys to know this is very important. Memorize it like your name. Tamam? Primary hemostasis, if we want to evaluate if it's working well or not, we do this through measuring the bleeding time. What is primary hemostasis? It's basically the formation of the platelet plug. So if we want to measure how well this primary hemostasis, how well these plugs are working, TTP, I think it's, no, this is for secondary hemostasis. We'll come to it, don't worry. But for now, you guys think of it logically. Tamam? If we want to measure how well a platelet is working, we're going to see how much time it takes for it to stop the bleeding. صح? Let's say a person is injured, within 2 minutes, the bleeding stopped. This is how you know when the platelets are working well. But what if someone is injured? We stayed 20 minutes where the bleeding didn't stop, not even by a bit. This is how you know there is a problem with the platelets, or there is a problem in the primary hemostasis. Okay? But if you're in a hospital, someone asks you, okay, we have a patient, we want to make sure his platelets are functioning well. How do you do that? Bleeding time. Measure the bleeding time. This is for hemophilia, right? Hemophilia is not about platelets. It's related to this lecture. We'll come to it later, inshallah. Wouldn't the results of the bleeding time be skewed since it's not entirely platelets but also vasoconstriction? You have a point, but don't forget, endoplatelets are what release thromboxane. Thromboxane is what causes vasoconstriction. So in one way or another, these platelets are what cause vasoconstriction also. Of course. طيب. فخلاص, just remember, in the primary hemostasis is evaluated through bleeding time. طيب. Any questions? ترى والله guys, we are almost done with the lecture, inshallah, the second lecture. Not done, but almost. طيب, I hope everything previously discussed was clear. You made this so easy. Thank you. Of course, of course. Wallah, I'm trying my best. Inshallah, you won't have to worry about this lecture ever in the exam. طيب, I have a question for you guys. Take your time, read it carefully, or let me know what you guys think the answer is. Okay, so everyone is answering B, B, B. Like, let's read the question and see. A 6-year-old girl is brought with a history of epistaxis. Taban, just for your knowledge, epistaxis means nosebleed. You don't really have to remember it, but just for you to understand. Her laboratory investigations show that hemoglobin is normal. White blood cells are elevated, which means غالبا there is an inflammation. Platelets are normal. Peripheral blood smear show that normal platelets morphology is there. Okay, there's kind of a grammatical mistake. I'll fix later. Bleeding time is more than normal. Prothrombin time is normal. Activated partial thromboplastin time is normal. So everything is normal except bleeding time. If it's taking a lot of time to stop the bleeding, where is the problem? In the platelets, which means what laboratory investigation we should do? Platelet function tests, as you guys answered correctly. Absolutely. When do we get to the clotting factors like factor 8 and 13? Wallahi, now. Now we're going to get to those. Oh, don't worry, they're very easy and simple. I think I figured out a way to make them simple, inshallah. So hopefully it won't take us a lot of time. Wait, the chat disappear. Why not D? طيب, I'll go back. D is factor 1 SA. It measures fibrinogen. احنا مو قلنا انه we have platelets and then we have fibrinogen. طيب, احنا right now we want to focus about the platelets. لأن platelets are what stop the bleeding at first. We said in the bleeding time is about platelets. Fibrinogen is later. It's later in the coagulation process. Protein S and protein C, we will talk about them, but they are basically anticoagulants. They are things that stop coagulation, but they are not very high yield. يعني. Why can't it be elevated levels of protein C and S? Wouldn't they also prevent platelet formation? That's a very good question. Usually, if protein C and S are abnormal, we would also see a problem in APTT and PT. The protein C and S are involved in the coagulation parts, the clotting factors, the intrinsic or extrinsic pathway, more than the bleeding time of platelets. Tamam? That's when protein C and S would be the object of concern, of course. طيب, secondary hemostasis. We said that secondary hemostasis has one goal and one goal only, to make fibrin. But to make fibrin, it's kind of a complicated process, tamam? Secondary hemostasis starts with extrinsic and intrinsic pathways. Why are they called extrinsic or intrinsic? The extrinsic pathway will use factors that come outside the endothelium, from the tissues that are surrounding it. That's why it's called extrinsic. The intrinsic pathway will come from stuff or factors that are found inside the endothelium already. Okay? Well, these two pathways will converge together to something called the common pathway. Well, this is the common pathway. It is the final part which will make fibrin. How is fibrin made? Wait, we have a question. Why do we need both intrinsic and extrinsic pathways? Don't they both just make fibrin? Proof, the thing is, intrinsic factors will contribute in some ways, extrinsic will contribute in other ways. They are both important. Basically, they are working together to achieve the same results. تمام? طيب, you guys will understand it. What I want you to know from this slide right now is in secondary hemostasis, the final goal is to make fibrin. Is this sentence clear? حلو. طيب, well, the first step of secondary hemostasis is two pathways that will converge together. Well, this is what's going to help us make fibrin. Is this sentence also clear? حلو. Let's talk about these two pathways that converge. تمام? As we said, we have the extrinsic pathway and the intrinsic pathway. Let's start with the extrinsic pathway. As we said, extrinsic, why is it extrinsic? It involves factors that are coming from outside the cell. Now you need to memorize the factors. Extrinsic involves factor 7, which is this one, it's in Roman numerals, and tissue factor. من اسمها tissue factor, tissues are found outside the cells, so it's going to be the extrinsic pathway. Tissue factor, which is also known as factor 3, will come with factor 7. Together, they will start the common pathway. This is all you need to know about the extrinsic pathway for now. خلاص, is this clear? Yani basically, I want you guys to memorize which factors are for which pathway. فبال extrinsic pathway, we have tissue factor and factor 7. Those two factors. Tissue factor is thromboplastin. It will help make it. Yes, absolutely. Can you please explain the difference between vessel injury and contact? Well, what do you mean by contact exactly? Look, vessel injury means the blood vessel is damaged. The blood vessel, خلينا نقول, it's damaged or you started bleeding. That's injury. Or contact can cause injury. What about calcium? طيب. Calcium, what it does is basically it helps all of these reactions happening in all the pathways, extrinsic, common pathway, some of the things in intrinsic. يعني زي ما انتم شايفين, calcium is literally mentioned in every single pathway. Well, I just want you to know that calcium makes everything possible. As simple as that. فإحنا we have tissue factor, which is factor 3. It will combine with factor 7. This is the extrinsic pathway. ويعطيكم العافية. This is what I want you to do. What do these two factors do? They will initiate the common pathway, which starts with factor 10. طبعا, how do we measure the extrinsic pathway? By measuring the prothrombin time. تمام? Keep this for now as a memorization type of thing. I will explain it in detail, but just keep it in the bottom of your minds for now. Let's talk about the intrinsic pathway. Intrinsic pathway. It starts with 12, and then 11, and then 9, and then 8. You have to memorize them. 12, 11, 9, 8. These are the factors that are involved in intrinsic pathway. In the intrinsic pathway, we don't have the tissue factor. Tissue factor is only in the extrinsic. لأنه من اسمها tissue factor. Tissues are outside the cells, so it's going to be for the extrinsic pathway. In the intrinsic pathway, we have 12, 11, 9 or 10. Well, basically, these are not very important, but in 12 will become 12A, but then will become 11, but then 11A, but then 9, but then 9A, but then 8. Okay? You don't really need to know the A's وما إيش. Just know that the factors that are found in the intrinsic pathway are 12, 11, 9, 10, 9, 8, sorry. 10 is found in the common pathway. Also, 10 is a cool number, X. It's basically the culmination of everything coming together. The common pathway starts with X. So intrinsic is when the vessel is damaged, and extrinsic is when there is tissue damage. That's one way to remember it. But generally speaking, when we have bleeding, more often than not, both of them will activate, both of them together. 12 will activate and give us 11, and it will give us 9, and then it will give us 8. Yes, that's true. So is tissue factor 3, or is factor 3 a type? لا. True, guys, in medicine, there are some things that have multiple names. ليش? لأنه a bunch of egotistical scientists couldn't agree on one name. فواحد سماها tissue factor, وواحد سماها factor 3. They are the same thing, the exact same thing. And both of them will activate 10? Yes. Both of these two pathways will come together to make the common pathway, which starts with factor X. How do we measure the intrinsic pathway? By measuring the APTT time. مو إحنا قلنا how does 9 and 8 make factor 10? Basically, they don't make it, they activate it. That's a better way of putting it. What's APTT? Okay, we're gonna come to it right now. شوف. PT is prothrombin time. I'll skip just for you to know. PT is prothrombin time. That's how we measure the extrinsic pathway's integrity. APTT is activated partial thromboplastin time. That's what it stands for. You don't need to know exactly what it stands for. I just want you to know in the APTT is intrinsic, PT extrinsic. There is a very, very famous mnemonic. This mnemonic, everyone uses it to memorize this. طيب. I want you to forget the A. We have PTT and PT. Play table tennis and play tennis. I want you guys to answer this question. Where do people play tennis, normal tennis? Do they play it indoors or outdoors? Usually. Outside. طيب, what does extrinsic mean? It means outside. But play tennis, PT, is going to be for the extrinsic pathway. You play tennis outside. But what about APTT? Play table tennis. Where do people usually play table tennis? They play it inside, indoors. But that's why intrinsic pathway. خلاص? So if you ever get confused, APTT, play table tennis, you play table tennis inside. But that's the intrinsic pathway. PT, which is play tennis, you play tennis outside, and that's the extrinsic pathway. That's how you memorize which one is for which. تمام? Of course, guys, ولو والله يعني واجبنا. I'm trying to make everything as simple for you, لأن I will admit this lecture is kind of heavy, but inshallah with this past session, it will be the easiest lecture for your FND final. طيب, some of you guys are asking what is APTT and what is PT? Do you guys remember how we said إنه to know if primary hemostasis is working well, we measure the bleeding time? The exact same concept applies here. Primary hemostasis, to know if it's doing well, measure the bleeding time. Extrinsic pathway, to know if it's doing well, measure the prothrombin time. PT, tennis outside. Intrinsic pathway, to know if it's doing well, measure the APTT, which is the play table tennis inside. تمام? طيب, now when it comes to the intrinsic pathway, someone asked about, I don't understand the PT and APTT label, sorry. طب listen يا من, شوف. In the next two slides, I have full descriptions about them. But let me get to those two slides, I'll explain them to you. For now, I just want you to memorize. In APTT measures how well the intrinsic pathway is, or PT measures how good the extrinsic pathway is. Do we have a deal? يعني if someone has an abnormal PT, معناتها we have a problem with the tissue factor or factor 7. If APTT is abnormal, يعني we have a problem with 12, 11, 9, and 8. Okay? Now, something I want you guys to memorize is that certain factors, if they are deficient, if they are not found in a person, they can cause a disease called hemophilia, which is excessive bleeding. مو إحنا the factors help us in making the fibrin. If they're deficient, we will have excessive bleeding. If factor 11 is not here, we will have hemophilia C. It's a kind of hemophilia. If factor 9 is absent, we will have hemophilia B. And if factor 8 is absent, we will have hemophilia A. Can we guys agree on this? Yani if you get a question in the exam saying, a child is diagnosed with hemophilia C, which of the following factors is deficient? You're going to tell them factor 11. Okay, that's basically why I'm telling you this concept. Okay, I'll repeat it. There are certain factors, if they are not found in a person, they will have a disease called hemophilia, which means bleeding, excessive bleeding. Tamam? Well, I want you guys to know which factor causes which kind of hemophilia. If we have a problem in factor 11, then we will have hemophilia C. If we have a problem with factor 9, then we will have hemophilia B. If we have a problem with factor 8, then it will be hemophilia A. Is the concept clear now? And best you need to know which type of hemophilia we will have if a certain factor is missing in a person. Tamam? That's basically all I wanted to know. Try to go down. What about for extrinsic? A problem in the extrinsic pathway would not cause hemophilia. It could cause different diseases that you guys don't need to know, but it will not cause hemophilia. تمام? Well, don't worry about PL. It's probably platelets, but what I want you to know here is 8. I want you guys to know in the intrinsic pathway, 12, 11, 9, 8. 11 hemophilia C, 10 hemophilia B, 11 hemophilia C, 9 hemophilia B, 8 hemophilia A. Extrinsic pathway is tissue factor and factor 7. Or another name for tissue factor is factor 3. All of these factors will help each other to start the common pathway, which begins with factor X. Is that clear? Yes, Noor is absolutely correct. Thank you for reminding me. Basically, in the question, غالبا they're going to tell you which of the following factors causes hemophilia C. But what if the question goes like, which of the following factors causes hemophilia, period? Full stop. Are you going to choose 11 or 9 or 8? Because you might be confused. All of them cause hemophilia. We're going to choose 8. You want to know why? Not because it's necessarily the case, because it's the most common. Okay? If you get a child in your clinic, will they tell you he has hemophilia? But we don't really have time to test which factor he's missing. In this case, you're going to have to gamble. Your best gamble will be factor 8, because it's the most common cause of hemophilia. That's why it's A, Aslan. It's the first hemophilia, hemophilia A. It's the one they discovered first. What causes the missing factors? Genetic mutations. Genetic mutations will cause the missing factors. Tamam? But just know in factor 11 deficiency is hemophilia C, factor 9 deficiency is hemophilia B, factor 8 deficiency is hemophilia A. Allah ya'afik, habib qalbi. Is that clear, guys? This is the intrinsic or extrinsic pathway. Yeah, I know, guys, and there is a lot of room for exploration with these topics. And what is a factor? What does it do? How do they come together? How do they activate? But you guys don't need to go into this detail. I told you exactly what you need to know. Extrinsic tissue factor, factor 7, they come together. And you measure them with PT time, which is play tennis outside. Intrinsic, 12, 11, 9, 8. Well, they are measured by APTT time, play table tennis inside. Factor 11, hemophilia C, factor 9, hemophilia B, factor 8, hemophilia A. But depending on which factor is deficient, you will know what kind of hemophilia we have. I hope this is clear. This is the intrinsic or extrinsic pathway. They both come together to start the common pathway. Well, the common pathway is what's going to end up with making the fibrin that we want. But we can't start the common pathway without the intrinsic or extrinsic pathways. Right. Now let's go into some more detail. Let's take every pathway on its own. We said when blood comes into contact with an injured tissue thromboplastin, someone asked, is thromboplastin tissue factor? Yes, it is, as you guys see. Tissue thromboplastin, it's basically another name for the tissue factor, which is factor 3. It's going to interact with factor 7. Well, those two together will make factor 10, اللي هو the common pathway. Another name for factor 10 is the Stewart factor. It's just another name, guys. Nothing too deep. As you guys can see here, damaged tissue, factor 3, tissue factor. Through calcium, it will be able to bind to factor 7. وبعدين they will give us factor 10. طيب. To measure the integrity of the extrinsic pathway, to measure how well it's doing or how well it's functioning in a person, we measure the prothrombin time. How long it takes to make prothrombin, to thrombin. It's a certain reaction. I'll explain why in a moment. But if this reaction happens in a normal time range, but you know, in the extrinsic pathway in a person is normal. If the prothrombin time in a person is way too long, then that's how you know we have a problem in the extrinsic pathway. It's unable to work fast. And we said prothrombin simulates coagulation, right? Absolutely. We'll know another function of it right now in a moment. Another function you're gonna know in the common pathway. Tamam? But guys, I just want you to make this link. Do you guys remember when we said if we want to know if the primary hemostasis, which are platelets, if we want to know they're doing well, what do we measure? I want someone to tell. Yalla, someone in the hospital told you we want to make sure this person's platelets are doing well. The primary hemostasis, which is responsible for the platelet plug, is doing well. How? Exactly, bleeding time. using the same logic, if we want to know if a person's extrinsic pathway is okay, we measure the prothrombin time. Same logic. With a prothrombin, we'll elevate and evaluate, the presence of the extrinsic pathway factors اللي هم 5 و 3 the tissue factor وكمان factors of the common pathway اللي هم 10 و 5 وهذول الأشياء. Do we measure it by injuring the patient? That's one way of measuring it, yes. Yeah, like a small cut. Is prothrombin the same as factor 2? Prothrombin is another name for Allahumma salli ala Muhammad. Factor 2, yes. This question popped up in an MCQ. If there is a problem with vitamin K and PT is long. Okay, you kind of predicted a question I was going to ask in the end of the lecture. Keep it for later. I will explain this exact concept to you that's related to vitamin K. Just keep it for the last. For the end. تمام? طيب. But let's say you get a clinical scenario where they tell you in the prothrombin time is too long. Which factors will be affected? I don't want you to tell me which pathway. We know it's the extrinsic pathway. But which factors are going to be affected? Tissue factor and 7? Yes, 3 and 7. 3 and 7, absolutely. Other than that, by the way, in the intrinsic or extrinsic, يعني if there is a problem, حتى the common pathway factors will be affected. But 10 too, yes, as you see here, 10 and 5. Because factor 5 or factor 10 are part of the common pathway. But to think of it logically, if we have a problem in the extrinsic pathway, we depend on the extrinsic pathway to make the common pathway, فحتى the common pathway will be affected. صح? Does that make sense? But yes, when we have, don't worry about calcium. Calcium is always found in the body. But I'll repeat. If we have an increased prothrombin time, it's too abnormal, too long. It means we have a problem in the extrinsic pathway, which is this one. Let's go back. This one. If we have a problem in the extrinsic pathway, it means the problems we have are in the tissue factor, اللي هو factor 3 and factor 7. صح? طيب, we said that the role of the extrinsic pathway is to begin the common pathway. But if we have a problem with the extrinsic pathway, wouldn't that mean the common pathway wouldn't be able to start properly? It would. It will be affected. فلأنه the common pathway will also be affected, the factors of the common pathway will be affected too. It would be 3, 7, 10, and 5 كمان. And all of the factors of the common pathway, 2, 5, 10, we will go through them in a bit. Is that clear? Okay, let's move on to the intrinsic pathway. Can we question? Okay, ممتاز. The intrinsic pathway, as we said, if we want to measure its integrity, if it's doing well, it's by measuring the APTT, activated partial thromboplastin time. APTT, play table tennis inside. How will all of them be affected? شوف, let me explain it once again. If we have a problem in the extrinsic pathway, the common pathway will be affected too. Then the common pathway, it's waiting for the extrinsic pathway to tell it starts. حتى intrinsic, the common is sitting in the middle, waiting for the extrinsic and the intrinsic pathway to tell it to begin. If there is a problem in the extrinsic pathway, how will the common pathway begin? It won't. So that's why in a problem regarding, I still don't know what's the PL. PL is probably platelets, the platelets that are already there. But you don't need to know anything about it, صراحة. I've never seen it in a question. We've never talked about it. Does it need both of them to work? Yes, it needs both of them. Both of them come together for it to work. If we have a problem with the extrinsic pathway, we will have a problem with factor 3 and 7. Because the extrinsic pathway regulates the common pathway, حتى the factors of the common pathway will be damaged. PL is phospholipid surface. Wallahi, maybe. PL phospholipid, wallahi, I'm not sure صراحة. But whatever it is, it really plays minimal role. Whether it's platelets, whether it's phospholipid, whatever PL is, it genuinely has no significance. The biggest piece of evidence is here مثلا in the intrinsic pathway ما في PL. يعني في platelets بس they don't really do anything. The intrinsic mechanism focuses on 12, 11, 9, and 8. That's all you need to know. Other name for thromboxane, yes, TXA2, that's the other name. طيب, for the intrinsic pathway, we measure how well it's doing by seeing the time of APTT, activated partial thromboplastin time. PTT, play table tennis insight for intrinsic pathway, which will evaluate the presence of 12, or 11, or 9, or 8, and the common clotting pathways. I told you guys, if you have a problem in the extrinsic pathway, هتكون في مشكلة بالextrinsic factors and the common factors. Problem in the intrinsic pathway, the problem will be in the intrinsic factors and the common pathways. Is that clear? Does that make sense to you guys? Okay, ممتاز. طبعا, remember how we were talking, common will be damaged both ways. Exactly. طبعا, guys, do you remember how we said in the intrinsic pathway, every factor can cause a certain hemophilia if it's not there? Factor 11, hemophilia C. Factor 9, hemophilia B. Factor 8, hemophilia A. Oh, I want you guys to remind me, which one was the most common? Which factor deficiency is the most common to cause hemophilia in general? Yes, factor 8. فلأنه it's the most common, they decided to call it anti-hemophilic factor. And if someone has factor 8, it's kind of خلاص anti-hemophilic. It's like they won't have hemophilia. Do you guys get that concept? That's why it's called anti-hemophilic. It's just a cool name for factor 8. Can you repeat the anti-hemophilia? All right. Proof. We agreed إنه the most common deficiency that leads to hemophilia is factor 8, right? Which means إنه factor 8 is the most important and most common when it comes to hemophilias. إنه if we get 100 hemophilia patients, 70% of them will have hemophilia A, which is a problem in factor 8. So because factor 8 is so important for hemophilia, they called it anti-hemophilic factor. Because if you have it, you don't have hemophilia. يعني أنا الحمد لله, you الحمد لله, inshallah, you don't have hemophilia. It means you have this anti-hemophilic factor, which is factor 8, all right? طيب, let's move on to the common pathway. Now the intrinsic pathway did everything it should. Extrinsic pathway did everything it should. Now it's the last, final, easiest step, which will finally give us this fibrin that we want. The common pathway. طيب, how can we activate fibrin? We need something called thrombin. Do you guys remember thrombin? We said that it activates platelets, so it helps in coagulation. Another function of thrombin is to activate fibrin. But thrombin in the body is found in an inactivated form, prothrombin. مو قبل شوي I told you guys if something ends with OGEN, it means it's inactivated. If something starts with pro, it also means it's inactivated. But at the end, can we please look at the vasoconstrictors and dilators and procoagulation, anticoagulation, من عيوني. We're gonna go through everything again. We're done. Don't worry. Prothrombin is found in the body inactivated. It's also known as factor 2. كذا قاعد in the body. Because if it's activated, it's gonna make a lot of fibrin. We don't want that. But in the normal scenario, thrombin is inactivated. It's prothrombin. Intrinsic or extrinsic pathway, when they begin the common pathway, they're going to activate something called prothrombin activate. Well, this is what will make the thrombin active. The thrombin will start working. Well, طبعا factor 5 is also involved in this. You don't need to know exactly what, just know in the factor 5 is part of the common pathway. But if we want to make fibrin, we need something to activate this fibrin. What will activate the fibrin? Thrombin. بس هذا الثرومبين هو نفسه he needs activation. How is he activated? By the merging of the intrinsic and extrinsic pathway, which will finally activate factor 2, which is prothrombin. Is this first part clear? Okay, you know what? We'll explain the full picture مع بعض. Our final goal, wait, let me use a pointing card. Our final goal is to make fibrin. What will activate fibrin? Thrombin. How can we activate thrombin? By the intrinsic or extrinsic pathways. They are the prothrombin activators. For the intrinsic or extrinsic pathways, when they're finally done, they will finally activate prothrombin and make it into thrombin. With this thrombin will turn fibrinogen into fibrin, which is the final thing we need. يعني حتى if you guys remember the very first slide, when you guys were showing lost about it, it was this one. We said with the extrinsic pathway or intrinsic pathway will come together to make prothrombin into thrombin. With this thrombin will finally make the fibrinogen, which is inactive, into the fibrin that we need to stop the bleeding. Is that clear? We said it starts with pro and it's inactive. Okay, look. If anything that ends with inogen means it's inactive. And anything that starts with pro also means that it's inactive. And this leads to secondary hemostasis. All of this is secondary hemostasis. Guys, this slide is literally secondary hemostasis in a nutshell. Extrinsic, intrinsic pathway. They come together. And they finally allow the activation of thrombin. With this thrombin will finally allow the activation of fibrin. This is the common pathway. We're going back here. Come on here. We can explain it here. Extrinsic pathway. Tissue factor and factor 7 start the common pathway. من الintrinsic عندنا 12, 11, 9, 8, they will start the common pathway. In the common pathway, we have factor 10 and factor 2, which is prothrombin. Basically, factor 10, with the help of the thromboplastin and the extrinsic and intrinsic pathways, the prothrombin will become thrombin. With the thrombin, will turn the fibrinogen, which is inactive, into the active fibrin that is used for clotting. I hope the full picture now makes sense. You're right, Abdullah. Yes, it's basically a second name for the prothrombin activating. Does the full picture make sense, guys? And everything linking with each other? Mumtaz. I'll still repeat it one more time to solidify it. Tamam? We have the intrinsic pathway, which is found inside the cells, or the extrinsic pathway. Is factor 5 used to activate prothrombin? Yes, it is used. Extrinsic pathway, intrinsic pathway. Extrinsic pathway involves the tissue factor, which is factor 3. Again, tissue because it's outside the cell for extrinsic, with factor 7. Tamam? This extrinsic pathway is measured through the prothrombin time, PT, play tennis outside. If the PT is abnormal, إيش يعني؟ معناتها we have a problem with the extrinsic pathway, not the intrinsic. Going to the intrinsic pathway, we have 12, 11, 9, 8. These are measured by the APTT, play table tennis inside. If the APTT is abnormal, then that means we will have an issue with the intrinsic pathway and not the extrinsic pathway. Tamam? Well, as we said, every factor in the intrinsic pathway, if it's not there, we will have a type of hemophilia. Factor 10 deficiency is hemophilia C, factor 9 deficiency is hemophilia B, and factor 8 deficiency is hemophilia A. And what was the most common hemophilia? A, through factor 8. Tamam? These two pathways, extrinsic, خلاص, they come together or they initiate the common pathway. The common pathway, the goal of it is to first make prothrombin into thrombin. Why? Why are we making thrombin? It's because this thrombin will make the fibrin we need. It will activate the inactive fibrinogen and turn it into fibrin. This is the common pathway. Is it clear, guys? Does both pathways occur? Yes, both of them occur. Thromboplastin plus factor 10 plus 5 plus calcium turn prothrombin into thrombin. And this step is the common pathway? Yes, Mariam, absolutely. Absolutely. After we have thrombin, the thrombin will change fibrinogen into fibrin. Does everything make sense? Tara, we're practically done with the lecture. That's why I want to take my time with this, just to make sure that you understand every single detail. What is 10 used for? 10, it will basically combine with 5 to make prothrombin into thrombin. Do we need to know anything about factor 13? I will come to it right now. But just tell me, until this point, is everything clear? Okay, alhamdulillah, alhamdulillah. طيب. Now, moving to the last thing in the common pathway. Fibrin is very strong, don't get me wrong. But there has to be something. Is there another name for factor 5? I'm not sure. But even if there was, you really don't have to know. It's irrelevant. Okay, some factors, maybe it's good to know their other names, but not factor 5. طيب. As strong as fibrin is, wait guys, give me a moment. I apologize. طيب. Thromboplastin is factor 3. You could say, yes, thromboplastin is factor 3. Yes, you can say that, which is the tissue factor. So we just need to know that it's factor 5. Absolutely. طيب. For this fibrin, خلاص, now we have it. It's very strong, by the way. But we need something that stabilizes it. Who's going to do that? The final factor of the lecture, factor 13. That's all you need to know about factor 13. Factor 13 causes cross-links between the fibrin to make it stable. تمام? This is factor 13. Stabilizes the fibrin by cross-linking. For the common pathway, once again, the first step is to activate prothrombin into thrombin. And that is done by prothrombin and calcium, which come from the extrinsic and intrinsic pathway. We already discussed this. Second step is to use this thrombin to make fibrinogen become fibrin, which is active. Fibrin has to be stabilized by factor 13 so it doesn't break. طيب, let's make a quick review before I say this last point. How do we measure primary hemostasis, platelet plug? How can we know it's doing well or not doing well? Bleeding time, absolutely. طيب, how can we know if the extrinsic pathway is doing well? What do we measure? PT, absolutely. But what about the intrinsic pathway? APTT, absolutely. طيب, I just want to add one more thing to this list. Factor 13, if we want to make sure it's working in a person, we do the clot solubility test. This test will determine if factor 13 is working ولا لا. تمام? That's the last test I want you guys to do. خلاص? Obviously, turning fibrinogen into fibrin depends on how fast thrombin is. You zoned out? ولا يهمك. I'm going to repeat the concept again, don't worry. Okay. We said in no primary hemostasis, we use bleeding time to test it. Intrinsic pathway, we use APTT. Extrinsic pathway, we use PT. Okay? If we want to measure how good factor 13 is in a person, we do the clot solubility test. Is that clear? Okay, alhamdulillah. طيب, by the way, guys, if both PT and APTT are prolonged, المشكلة is in the common path. Thromboplastin plus factor 10 plus factor 5 plus calcium turns prothrombin into thrombin, and this step is in the common pathway. Can you re-explain this? Okay, we can go here. We can use this one. After the extrinsic pathway or the intrinsic pathway finally activates factor X, this factor X will combine with factor 5 to turn prothrombin into thrombin. Is that clear, Mariam? That's basically it. What's in the common pathway, as we said. That's basically it. Of course. طيب, as we said, if PT alone is the problem, معناتها المشكلة is in the extrinsic and common. مشكلة APTT alone, a problem in it, then the problem is in the intrinsic pathway and the common pathway. What if both PT and APTT are abnormal? This means in the common pathway كله خربان. It's an issue in the common pathway. If the problem is in PT or APTT. If the problem is in factor 13, things will be normal. To test factor 13, you have to do the clot solubility test. خلاص? طيب, now that finally the body recovered from the injury, we don't need the fibrin anymore, we don't need the platelets anymore. We're going to break down the fibrin وخلاص. Do you guys remember what breaks down the fibrin? Exactly, plasmin. So the tissue plasminogen activator, it will turn plasminogen, which is inactive, into plasma. This will lyse the fibrin clot. In the body, we have plasminogen sitting there doing nothing. When the body sees that there is a clot that has to be broken down, the tissue plasminogen activator, again, it's an activator, it will turn the plasminogen into the active plasmin. Well, this is what will break the clots. تمام? طبعا, it's good to know when a plasminogen can be produced by eosinophils. Plasmin can also be produced by a bacterial enzyme called streptokinase. طبعا, guys, you don't need to go into detail with this, but I'll just tell it to you to feed your curiosity. Do you guys know why this is important? Whenever we have like a bacterial infection or an injury, مثلا bacteria invades, you guys know why it's dangerous. And sometimes when you get an injury, they always tell you, be careful of an infection, be careful if bacteria invades. It's because bacteria, they have an enzyme called streptokinase. Streptokinase makes plasmin. If you're bleeding and there is streptokinase, the moment your body starts making clots, this streptokinase will make plasmin, which will destroy these clots, which is not good. You're going to keep on bleeding and bleeding and bleeding. The wound won't heal. Exactly. Again, you won't be tested about this. I'm just telling it to you for the sake of your knowledge. Yeah. I just want you to know when the plasmin is what breaks the fibrin, or the tissue plasminogen activator will make plasminogen into fibrin. Plasmin, sorry, which will break the fibrin. تمام? طبعا, our bodies بصفة عامة, they have a lot of anticoagulants, natural ones, which prevent intravascular clotting. What do you mean break fibrin? Okay, let's go all the way here. Sorry. طيب, مو احنا قلنا the final step of stopping bleeding is to make this fibrin. We don't need this forever. Eventually, the blood vessel will regenerate through angiogenesis. It will regenerate. The injury will no longer be there. فخلاص, we don't need the fibrin anymore, so we break it down. When will the recording be posted? يعني مفروض a few hours right after the class session, inshallah. Of course. Right, خلاص ترى guys, والله we're almost over. Just one more concept, inshallah. We'll be done with both of these lectures. This is all you need for these two lectures. Come on, I think you have a lecture by Dr. Raihan about this, the extrinsic, intrinsic pathway. This should be enough for that too, man. I just want you to know in our bodies, naturally, they have a lot of anticoagulants. عشان inside the vessels ما يصير في clotting. Some of them are heparin, antithrombin. It makes sense, antithrombin. Or protein C or protein S. These are anticoagulants, basically. That's all you need to know. طيب. Last but not least, this one is for Munib, vitamin K. Proof. There are 4 factors in the body that need vitamin K to be activated. All the other factors, معلش, they're doing fine. But there are 4 factors in specific that need vitamin K in order for them to be activated. These factors are 2, 7, 9, and 10. I want you guys to memorize these factors like your names. يعني حتى if you guys notice, 2 and 10 are part of the common pathway, 7 extrinsic pathway, 9 intrinsic pathway. You have to memorize them. Vitamin K equals factors 2, 7, 9, 10. Very necessary. If a question comes and they tell you, we have a deficiency of vitamin K, which of the following factors will be affected? You're going to tell them 2, 7, 9, and 10. But deficiencies of vitamin K will result in non-functional factors. These factors will not be able to participate in coagulation at all. But how can vitamin K deficiencies happen? Maybe hepatitis, which is a liver disease, liver inflammation. Maybe it's the inability to absorb fats. That's how you can have vitamin K. Vitamin K basically dissolves in fat. Well, that's Crohn's disease. I want you guys to remember that Crohn's disease causes a deficiency in vitamin K. And if someone is using vitamin K antagonists, they're basically medications. You don't need to know much about them. I want you to know two things from this slide. Vitamin K associated with factor 2, 7, 9, and 10. Number two is that Crohn's disease will cause a problem in these factors. Is this point clear? And if anyone has any questions, let me know. Then the lecture is officially over. But for the remainder of this time, I just want to take your questions. It can be dietary. Is your PowerPoint enough to study for these two lectures? Salaha, inshallah, it's enough. Inshallah, it is enough. In any PAL session I try my best to make sure it's sufficient. I don't understand how vitamin K activates the clotting factors and the anti-clotting proteins at the same time. Truth, it's not that vitamin K is a procoagulant or an anticoagulant. It's just that the structures of these molecules need to be carboxylized. Carboxylation. Hello? There's a lot of molecules in the body that need carboxylation. The molecule or the substance that does this carboxylation is vitamin K. The factors 2, 7, 9, 10, they need carboxylation. حتى protein C and S, even though they're anticoagulants, they also need Allahumma salli ala Muhammad, this carboxylation. Yes, we will take a look at those. I just want you guys to answer the practice questions, وبعدين we'll immediately go to repeat anything you guys want. with you guys. Is this carboxylation so that the positive calcium can bind to them? That's one of the things. Yes, it's one of the things that we need carboxylation for. But generally speaking, no carboxylation means factor 2, 7, 9, 10 will be inactive. Tamam? Can we please look at the vasoconstrictors? Yes, I didn't forget about this. Inshallah, after the practice questions, we're going to go back to those. I did not forget. I don't fully get vitamin K affects the whole process at the same time. It's not really that vitamin K affects the whole process. It's just that you have certain factors that need to be activated by vitamin K. Why exactly? There isn't really a certain reason, but that's why I'm telling you, you just have to know them by heart. And the factors 2, 7, or 9, or 10, they need vitamin K to be active. That's pretty much the whole point. Tamam? I hope this was clear. Well, guys, just to clear out the confusion, I'll show you a question. طيب, I want you guys to focus on this question, tamam? Before rushing to answer, they said there is Crohn's disease, but not only Crohn's disease. The PT and the APTT are long. We have two issues here. Issue 1 is Crohn's disease. Issue 2 is abnormal PT and APTT. So you have to remember what factor will be affected with Crohn's disease, what factors will be affected with PT and APTT when they are both abnormal. Or add the factors together or tell me what you get. Okay, mashallah, everyone is saying C. Yeah, C is the correct answer, mashallah. It is absolutely C. Well, guys, genuinely, this is probably the hardest question you can get in the foundation final. Like, genuinely, for the fact that you guys were able to answer this is amazing. This is a question that came in previous exams, used to drive us crazy, but the fact that you guys got it correctly, mashallah, is an amazing sign. Is 5 the extra one? Yes. Why is factor 5 involved? Let me tell you. Let me switch to the pen. They said there is Crohn's disease. صح? What is Crohn's disease? Crohn's disease is related to vitamin K, which means the issue is in factors 2, 7, 9, oh my God, horrendous handwriting, 9, and 10. صح? Crohn's disease is basically inability to absorb fat, which will lead to a vitamin K deficiency. So these factors will be affected. So you might be asking, then why isn't B the answer? It's because you have to continue reading the question. They said there is an active Crohn's disease and the PT and APTT are abnormal. PT, APTT are prolonged. Do you guys remember when we said, let me go back to the pointer, what we said here, if both PT and APTT are prolonged, we have a problem in the common pathway. What are the factors in the common pathway? 10, 5, 2, those ones. But that's why in this question we have two problems: Crohn's disease or an abnormal PT-APTT, which means an abnormal common pathway. But the answer would be 2, 7, 9, 10 for the vitamin K issue, and 5 for the common path. Tamam? If vitamin K was in the options, would it be correct? Shoof, it would be correct, because they wouldn't put it as an option with option C, and both of them are correct, Wouldn't Crohn's disease make both prolonged anyway, since factor 7 and 9 are in the intrinsic and extrinsic pathway? I get your point, but not really. لأنه when we have a problem in the intrinsic pathway, it means all of the factors are not working well. But in this case, it's one of the factors اللي هو 9, Option B is the same as vitamin K, right? Absolutely correct. In practice questions, they mentioned mucosal bleeding. Is it solely related to von Willebrand disease? I'm not sure, to be honest, but probably is. It is probably related. But in this question, Crohn's disease, as we said, it's the disease where you're unable to absorb fat. Well, the reason why this is bad is because vitamin K is stored in fat. If you can't absorb fat, you can't really have the vitamin K. Or if you don't have vitamin K, you're going to have problems in factor 2, 7, 9, 10. Well, because we have abnormal APTT and PT, which are the common pathway, factor 5 will not be affected. Did AJR tell us this? Which part exactly? If you're talking about this question, then yes, 100%. And this question is actually a question that was found in past exams. I feel like he missed so much. Wallahi, I don't know, Salaha. I obviously did this past session based on his slides. I don't know what exactly he said, but that is exactly why I'm telling you guys for these two lectures, these two Dr. Abdul Jabbar lectures, just depend on this past session and on my slides, so inshallah it'll be fine. Okay, let's move on to the next question. Okay. Absolutely correct. The answer is, in fact, D, factor 13. As we said, the fibrin is strong in itself, but we need to help this fibrin attach with each other and attach to the site of injury by cross-linking. That's what factor 13 does. Yeah. Another question, mashallah, you guys got correctly. Another question. Wallah, you're going to get a question like this in the exam. ما فيها يمين يسار. This exact kind of question, you're going to get it in the exam. But if you answer this correctly, you don't have to be worried about the exam, inshallah. So as you guys can see, bleeding time is normal. Prothrombin time is also normal. What's abnormal is the APTT, activated partial thromboplastin time. It's abnormal. For which factor is deficient in this case? We have شوية conflicting answers. Before we answer, do we all agree that the problem here is in the APTT, which is abnormal, as a range? Do we agree that's what's abnormal? شوفوا انتوا bleeding time it's 5 والnormal is 1 to 6 that's normal. Prothrombin time is 10, normal is 10 to 14. The abnormal is the APTT. What did we say APTT is used for? Play, table, tennis, inside. What does it matter? Which pathway? Intrinsic or extrinsic? Intrinsic. I want someone to tell me all of the factors of the intrinsic pathway. What are they? 12, 11, 9, 8. Ya salam alaykum. But of those four, which one of them are found in the options of the question? Exactly. 8, which is A. This is how you want to approach these questions. Determine which one is abnormal. Think, are we playing tennis or are we playing table tennis? Extrinsic or intrinsic? Or then decide what to choose. Is this question clear to everyone, guys? This is how you want to approach this question. طيب طيب الحمد لله. The question اللي بعده. Okay, this is an easy question. Yeah. Can factors affect platelet count? Not really. The platelets are not part of secondary hemostasis. They're part of primary hemostasis. ما لهم دخل يعني. Platelets, we measure them by bleeding time, as we said, in primary hemostasis. Like, which of the following procoagulant protein binds to tissue factor, which is factor 3, and initiates the clotting cascade? اللي هو extrinsic pathway. Yeah, absolutely. The answer is C. 7 and tissue factor come together, they start the extrinsic pathway. طيب, this question. Which of the following is directly responsible for the lysis of insoluble fibrin? Okay, if someone wanted to argue with you guys and they were like, I think B is the answer, what would you tell them? Why is it not B? It's the inactive form. Exactly, exactly. يعطيكم ألف عافية. This is the exact answer. طيب, this was the end of the session. All of the questions, everything. But now I'm going to say, take any questions you guys have. لأنه honestly, يعني, a lot of new concepts in this lecture. It's one of the heavier ones. So if anyone has questions, feel free to say, we're going to explain everything. Other than that, we're done with the session. طيب, can you explain why A? طيب, in this question, they're asking what is directly responsible to break down the fibrin. إحنا قلنا إنه plasminogen is inactive. Plasminogen becomes plasmin. Or plasmin, it's the thing that directly breaks down the fibrin. So that's why the answer is fibrin. They said direct. Is that clear? Of course, guys. ما سوينا إلا الواجب. Well, I hope you guys do well, inshallah, on the exam. Inshallah, it will be easy. This PAL session is everything you need. Do we have PAL sessions for more? Yeah, you will have PAL sessions for more, inshallah. Made this so easy for us. Of course. Anytime, Allah. If you guys need anything, don't ever hesitate to contact me. Even if you have questions after we're done with the session. طيب. If the question asks for indirect, would the answer be B? No, not really. They won't tell you indirectly. And what would they tell? What they would tell you is which of the following is the inactive molecule that breaks down fibrin. That would be plasminogen. طيب, the first question. Let's go to 5. Yes, Ahmed, it's because they mentioned in both PT and APTT are abnormal, so the common pathway is affected. Exactly. طيب, let's go to the question of the vasodilators or vasoconstrictors. طيب, ثواني. Okay. Vascular tone. This one. We have a blood vessel. تمام? We said in what lines the blood vessel is the endothelium, this layer of endothelial cells. These endothelial cells can either widen the blood vessel or narrow it down. If we want a lot of blood to flow, then we dilate it. That's vasodilation. If you don't want a lot of blood to flow, مثلا when you're bleeding, we don't want a lot of blood to flow. That's when we do vasoconstriction. I want you to know in the nitric oxide does vasodilation. EDHF, endothelium-derived hyperpolarizing factor, does vasodilation. We have things that do vasoconstriction, like endothelin, you know. These are just examples of molecules found in the endothelium. Well, I want you to know when they cause either vasoconstriction or vasodilation. Let me make sure I covered everything about your question. Okay, procoagulation and anticoagulation. We're gonna go to this. I made it in a table for you guys so you can memorize them at once. Antithrombogenic and thrombogenic. Antithrombogenic meaning we don't want clotting and we don't want coagulation. Thrombogenic means we want coagulation, we want clotting. We want clotting when there is an injury. When there is an injury, the von Willebrand factor will be exposed. The tissue factor will work. The plasminogen activator inhibitor will work. Why? Because we don't want to activate plasmin. We want to inhibit it. Plasmin will break down the fibrin. But in a thrombogenic state, if we want fibrin, if we want to help the body recover from the injury, we don't want the plasmin. We want the fibrin to stay. So these things are thrombogenic, basically. Wait, what did you put the stars for? Okay, it wasn't me who put the stars, to be honest. I'm not sure what they represent exactly, but don't worry about them, yani. How about we just memorize the 3 thrombogenic ones and whatever else is thrown at us, we assume is anti. That is obviously a strategy you can use in many lectures. But yani, the anti-thrombogenic, they're not hard. They make sense. Yani, شوف, plasmin, it breaks down fibrin. Yani, أكيد it's going to be anti-thrombogenic. They want to end the coagulation. Antithrombin is literally antithrombin. Nitric oxides or prostacyclin, we agreed they're important. They inhibit platelets. Tissue plasminogen activator is what activates plasmin أصلا. فthey make sense, طيب, is in the question before the last we chose 7 because they mentioned tissue factors and that could only be found in the extrinsic pathway 100%. If we go here, they were like, what activates the tissue factor? It's 7. 7 binds with 3 in the extrinsic pathway. تمام؟ طيب. Thrombinogen? لا. The inactive form of thrombin is called prothrombin. All right? This question popped up in an MCQ and I was confused if there is a problem with vitamin K and PT. Would the problem be with the factors of this? Okay. If we have a problem with vitamin K, then immediately we have a problem with 2, 7, 9, 10. PT is long. It means the extrinsic pathway is not working. يعني factor 3 or factor 7. Oh, no worries, Munib. خلاص, أصلا we're done with the lecture and I'm just taking questions so I can explain it to you. But let's go to the question أصلا itself. This was the question, تمام? This is what you can see in the exam. It's telling you that the patient has Crohn's disease. Crohn's disease basically leads to vitamin K deficiencies. But Crohn's disease means we're going to have problems in factors 2, 7, 9, and 10. Tamam? They also said this patient has long PT and long APTT. So here we have 2 issues. We have an issue that is Crohn's disease affecting the vitamin K factors 2, 7, 9, 10. And we have an issue in both PT and APTT. And we said if both of them are prolonged, then it means the common pathway is affected. So which factors would ultimately be affected? 2, 7, 9, 10, and the factors of the common pathway. So that's why here the answer was C. I think this was your question, sorry. Okay. Like, do we have any other questions? Anything at all? Well, again, if you guys are studying the lecture again or watching the past session from a recording, if you guys have any questions, please don't ever hesitate to contact me. Inshallah, I'll help you as much as I can, whether it's related to the past session or outside the past session here. I'm sorry, but can you fastly mention the common factors? Of course, yalla, let's go for them. Like, we said in extrinsic, 3 and 7. Intrinsic, 12, 11, 9, 8. Common would be 10 and 5, and prothrombin, which is 2. 10, 5, 2. These are the common. What was the role of factor 5 again? Okay, factor 5 will help factor 10 in making prothrombin into thrombin. It will help in the formation of thrombin. That's what factor 5 does. Well, this thrombin will then finally activate the fibrin that we need. As we said, factor 13 has only one role. It's to cross-link the fibrin to make it strong. Oh, guys, yalla, question for you. I'll reverse the question onto you. What is the test we use to make sure factor 13 is okay? Don't worry, Mohammed. I was just taking questions. We're pretty much done with the lecture. Hi, guys. What was the name of the test? Exactly, clot solubility test. That's the name of the test that determines the integrity of factor 13. I just asked Indo what test is used to determine if factor 13 is abundant and doing its job. So we said factor 13. Is it included? Shoof. Yeah, it is included. What's included in the, not in the common pathway, yeah, kind of in the common pathway, it's something on its own, which basically cross-links the fibrin to make it strong and stabilize. But yes, it is included. You have to know it. They always ask about it. I can guarantee you're going to see a question about it. Is 13 similar to 8? No. Factor 8 is in the intrinsic pathway. We have 12, 11, 9, 8. This is factor 8, which factor 8 is related to hemophilia A, as we said, the most common hemophilia, the anti-hemophilic factor. Factor 13, اللي هو هنا, is only responsible for one thing, making cross-links in the fibrin for it to be strong, basically. Vitamin K affects 2, 7, 9, 10. Exactly, yes. It affects 2, 7, 9, and 10. Absolutely. Absolutely. Of course, wallahi موجودين anytime, inshallah. You guys have the slides. The session will be uploaded as a recording. Again, if you have any questions, please let me know, whether it's now, whether it's after the session. Well, yeah, I'll say a few more minutes just to see if anyone else has any questions. But other than that, we are officially done, yeah. Inshallah.